All right, welcome back, everyone. We're going to continue with our reproductive system material. Where we left off, we were just discussing the anatomy of the male reproductive system. Again, at the end of the last video, I was underlining a lot of different structures. A lot of these structures here we do need to be prepared for our final test as far as reproductive, but not everything. So just pay attention to the stuff that I was focusing on. Again, one big distinction is that the Quizlet does not show this, only because there are two diagrams we need to know for this. We do have the male and female reproductive systems, so just be prepared for that. And one thing I did want to clarify, I had some students reach out to me, so I did edit it in as a class comment. Our test is the 19th, not this coming Friday, but it's a Tuesday, a little ways away, just because I think it's going to take a little bit of time to make sure we complete this, we have enough time for review, et cetera, et cetera. Okay, so let's continue. So where this all begins, we essentially can separate it into a couple of major structures. We have the testes, which of course are going to be the male gonads, and then we have the duct system. As far as the duct system is concerned, that's going to be the epididymis, the ductus deferens, and the urethra. These are essentially just tubes. We talked about ducts back in endocrine system. We mentioned how for exocrine, which is a little bit different, it requires the use of these ducts in order for you to secrete what you need to secrete. Similar situation here when it comes down to sperm and the way that sperm travels. Now, there are some accessory organs. So these are organs that help out. Remember, we talked about accessory organs back in digestive system, how we mentioned how there are organs such as the teeth, such as the tongue, the liver, the gallbladder, that help with the digestive process. Well, these are organs that help with the male reproductive system. So what are these organs? We have the seminal vesicle, the prostate gland, the bulbourethral gland, and then the external genitalia as well, such as the penis and scrotum. So we'll get through all those as we continue progressing, but we got to start off somewhere. So we're going to go with the testes. Let me go ahead and reload this real quick just to make sure that all the slides are loaded in properly. But yes, we are going to start off with where the sperm is made, in the testicles. Now, in the testes, we have both men have two sets of testes, or two testes, one set overall. It is a situation where if one of them happens to get damaged via like torsion, for example, which is a twisting action, or testicular cancer, which is something that is very serious that can affect men, you can still procreate with one healthy testicle. But obviously, it does limit and lower substantially your sperm count. There is a reason why men have a substantially high sperm count. As we'll learn in this unit, per ejaculate, there are millions with an M, millions of sperm. And that's by design. You need those type of numbers in order for procreation to even occur. So yes, if you do have a situation where one testicle is removed, it is very possible for procreation to still occur, but it does lower your numbers and possibly increase or decrease the chance of successfully implanting with the ova. So that is something to be aware of. Now, as far as some of the tissue that we see here, we have these special coverings. I like this picture just because it kind of gives us like a clear like X-ray version of what you would see. First, we have what's called the tunica albuginea. Basically, I don't get too crazy with this with regards to this unit, but it is a covering that just covers the internal testicle tissue. So all of this is inside the scrotum. All of this region right here, this is all inside the scrotum. But we have this white layer that's called the tunica albuginea. Again, we don't need to get too crazy with that, so we're going to continue moving forward. A little bit more as far as the covering of the tissue, we have what's called the septa or the septum in plural, extensions of the capsule that extend into the testes and divide it into lobules. So what we see here, just to give us a little bit of underlining, again, all that white tissue was just a tunica, which I'm not too crazy about having to focus too much on. Then we see the septum or the septa, which are these little gaps that we see right here. And then finally, when you have all this stuff coiled up together, that's more specifically referred to as the lobule or the lobule. To me, funny enough, it just kind of looks to me like packs of ramen that have been kind of crumpled up together in this little region. Again, we don't get too crazy with these three terms, but other terms on here we do need to get a little bit more in depth with. So just wanted to share that with us. Now, if we continue progressing down, we'll see here that each lobule contains 1 to 4 seminiferous tubules. That is a term we do need to know. So the seminiferous tubule, I'm just going to underline that for us here. The seminiferous tubule is these tiny, tiny little tube structures. And what you see on the next slide, just to go back down, is that there are multiple of these seminiferous tubules found in these contained regions. These tightly coiled structures, they function as sperm-forming factories. So if on the test I were to ask you where specifically are the sperm made, that is going to be the seminiferous tubules. It is too vague to just say testicles, or the gonads for that matter. It is specifically the seminiferous tubules. They're going to empty the sperm into a structure known as the reti testes. You can see the reti testes right here. So I'm going to scroll back up a little bit and just kind of color or circle the region that is considered the reti testes. It is this discolored, palish color region where it goes from the seminiferous tubule, so more specifically the gonad or the testicle, and then it starts to enter this comma-shaped structure, which we're going to learn about shortly, known as the epididymis. So this is like an in-between region. All right. Interstitial cells produce androgens such as testosterone. So interstitial, you can break that word down into its components. Inter and then stitial. Remember, stitial was used to describe tissue. So literally, this is just cells found in between tissue. Those cells found in the testes are going to be responsible for creating sex hormones, which we learned back in endocrine system, otherwise known as androgens. And in this case, their major product they're creating is testosterone. Testosterone plays a vital role in the health of the male body, and we'll learn more about that as we progress. We did talk about that previously. We kind of previewed it back in endocrine system, but we'll get more into that as we continue. Okay. The epididymis. So let me get back up here. I'm going to be going through this a couple times just to show us more specifically what we're looking at. The epididymis, as they refer to it, is this comma-shaped structure that we see here that sits on top of the testicles or the testes. You have 2 of them, right? So if you have 2 testes, you have 2 epididymis. They're just sitting on top of them, as we see. So let's discuss a little bit more about what the epididymis does. The epididymis is this comma-shaped, tightly coiled tube. It is found on the superior part of the testes and along the posterior lateral side, as we mentioned, functions to mature and store sperm cells. So let's go ahead and underline that or circle this term, this entire bullet right here. That is very important as far as the major purpose of the epididymis. Why do we have this structure in the first place? It's right here. You want to mature and store sperm. So when the sperm are made in the seminiferous tubules, which are found in the testes, they are made as immature cells, as we call them. Those cells are not capable of reaching the ova and causing a fusion. It would never happen. Those cells have to stay where they're at in the epididymis, in this case, and become more mature. And when we talk about more mature, we're not talking about like emotional maturity that, animals like us experience. We're talking about literal growth. And in this case, growing things like the tail, which is the propulsion method for the sperm to swim and move. So all of this is necessary. Now, what we see here, it doesn't take necessarily 20 days, but it does store the sperm for up to 20 to possibly up to 30 days. So roughly like a month. So the question then becomes, what happens to that sperm if there is no expulsion? If the sperm stay in the epididymis for a month, what happens? Well, just like other body cells, if your body's not using those cells for its purpose, which literally the entire purpose of sperm is just to fuse with the ova, then your body recycles it. those sperm cells become old. Your body recycles those old sperm cells, and you use those products to make new sperm cells. So it's a very efficient process where you're able to consistently recycle them and hopefully have fresh, new, strong sperm in the case of when they're needed. Okay, this is going to expel sperm. So when they are going to exit the body, it's going to expel the sperm with the contraction of muscles in the epididymis walls to the vas deferens. So let me go back up here one more time. Near this area, you can't really see it in this picture, but this whole tube that we see here is called the vas deferens, connects to the epididymis. Surrounding that area, still in the scrotum, you have a very, very, very thin layer of smooth muscle. And that smooth muscle, as we learned, is involuntary. So there is no control here, but it's going to move and force the sperm towards this tube here called the vas deferens, or the ductus deferens is another name that we see. So speaking of, let's talk about the vas deferens. So I'm going to go ahead and just come back up here and just show us specifically what that looked like. So if we had the comma-shaped structure before, the vas deferens is going to be everything moving up in this direction. It is just a long tube that makes its way from the scrotum towards more of the membranous portion, as far as away from the scrotum. So that's literally its entire purpose. It is pretty convoluted, as you can tell, but it has to just travel and send the sperm away from the scrotum area. So let's read what we see here. Carry sperm from the epididymis towards what we call the ejaculatory duct, that is close to the prostate gland, if you remember our material from yesterday. Passes through the inguinal canal and over the bladder. Inguinal is a region of the body that's basically near your groin. We don't need to worry too much about that, but that's kind of the name, the general area. It moves sperm via peristalsis. Hey, that's that whole smooth muscle process, right? Where you can slowly move things in one direction just by controlling the smooth muscles and how they move and how they function. And then finally, there's this thing called the spermatic cord. I'll show you what it is when I scroll back up in just a second. It is the ductus deferens, also known as the vas deferens, blood vessels and nerves in a connective tissue sheath. So if we go right back up here, the spermatic cord is going to be all of this region. Basically, at the base, or where the scrotum begins, there is a cord-like structure. So it kind of coils it all together, as you see here, if you look a little bit under it, where all the blood vessels that nourish the scrotum and the testicles, more specifically, are coming in this direction. The nerves, of course, that send information coming in here directly, and the vas deferens. Now, the vas deferens, though, the key thing here is that the direction of the sperm is always going in one way. It's always going out of the scrotum. There is no sperm that's traveling back down towards the scrotum. That doesn't work that way. So it is always a one-direction thing when it comes down to the vas deferens. So that is something to clarify for us there. All right, so let's go back down. All right, a little bit more about the vas deferens. It ends in the ejaculatory duct, which unites with the urethra. So again, where it's going to end, the ejaculatory duct, which is connected to the prostate gland and essentially the urethra at this point. There is a term that a lot of you have likely heard of before. It is a surgical procedure that a lot of men, especially later in life, the average age is typically somewhere around 45 plus. This is in no way, shape, or form something that is regular routine. It's more so that a lot of men do this because they may not want to have any more children in their lives, if they already have one or two or multiple children. But a vasectomy. The act of a vasectomy is the cutting of the ductus deferens at the level of the testes to prevent transportation of sperm. Something super, super interesting as far as vasectomies are concerned. A couple fun facts for us to share. So originally, this surgical procedure was literally with like a small surgical scissors. You cut the vas deferens and you're done. If the tube cannot connect, the sperm will never make it nowhere near the penis, for that matter. Nowadays, this procedure can be done with like a rubber band type tissue. It's obviously not just a traditional rubber band, but what they do is they actually grab, like, let's say, for instance, this area right here. They would actually grab a region of the vas deferens and fold it in over itself. That folding in process prevents, it still does the same job. It prevents the sperm from flowing, from going any further. So you still accomplish what you need to do. You're preventing any sperm from passing through. But the biggest thing about this is by just folding it in over itself. In an instance where someone were to get a vasectomy, maybe a year or two later, they decide, oh, that was a mistake. I actually do want more children. You could get this procedure removed. You could do a reversal. So that is something interesting. Another interesting fun fact relating to vasectomies is it is not 100%. Just like condoms are at a 99.9% rate of preventing an unwanted pregnancy, vasectomies are basically in that same boat. There have been numerous situations where a man has a vasectomy, thinks, okay, it's impossible, but things still happen. There can still be accidents that occur. At worst, it's going to, as far as like not necessarily a botched surgery or just not getting the performances or the results that you would expect. At worst, you're just going to have a situation where instead of an ejaculate having millions of sperm, you're only going to have, thousands of sperm, which that substantially drops the number of sperm. And in a realistic situation, you could repeat that scenario hundreds of times. It's never going to reach the ova, but all it takes is one, right? So it is a situation where sometimes there have been cases where people do get these procedures done, and unfortunately they don't exactly get the results that they were looking for. Okay. And finally, we do have a little bit more about the urethra, but I think we're going to stop here. We just have to talk a little bit more about where everything's going to exit. And then I have a great mnemonic to remember the path that sperm takes. So we'll get into that into our next video. So no worries with that. But I appreciate you guys checking in for today. All right, I'll see you on the next one.